Mechanical ventilation triggers hippocampal apoptosis by vagal and dopaminergic pathways. Intubation and positive pressure ventilation increase the incidence of delirium by up to 74% to 83% compared with 20% to 48% in non-intubated patients. Critically ill patients who undergo long-term mechanical ventilation show distinguishable neurological impairment, including memory and cognitive decline. Mechanical ventilation is a known risk factor for these impairments, but the correlation between lung stretch and brain injury is unknown. Patients with delirium show brain structure alterations after discharge and even months later. The study helps provide evidence linking the two and may explain the neurological changes in ventilated patients, thereby identifying therapeutic approaches. Mechanical ventilation could directly cause brain injury by afferent neural signaling. Testing this hypothesis involved studying alterations in the brain areas using mice. The pathways leading to brain damage and treatment were also studied. If cells are no longer needed, they commit suicide by activating an intracellular programmed cell death. In adult tissues, cell death exactly balances cell division. If this were not so, the tissues would grow or shrink. Rather than dying due to injury, cells that go through apoptosis die in response to signals within the body. When cells recognize viruses and gene mutations, they induce death to prevent the damage from spreading. When a cell is compelled to commit suicide, proteins called caspases go into action. They break down the cellular components needed for survival, and they spur production of enzymes known as DNases, which destroy the DNA in the nucleus of the cell. The cell shrinks and sends out distress signals, which are answered by vacuum cleaners known as macrophages. The macrophages clean away the shrunken cells, leaving no trace, so these cells have no chance to cause the damage that necrotic cells do. Cleavage of PARP1 by caspases is a hallmark of apoptosis. The hippocampus is responsible for processing of long-term memory and emotional responses. Alzheimer's disease, a disease that affects elderly people and often results in memory loss, has been proven to have affected and damage to this area of the brain. The hippocampus is one of the first regions of the brain to suffer damage. Short-term memory loss and disorientation are included among the early symptoms. Hippocampal dysfunction might produce an alteration of dopamine release, affecting the communication of information to the prefrontal cortex. Dopamine is a neurotransmitter and may also serve as a neurotoxin and thereby participates in the neurodegenerative process. Transmits signals between nerve cells in the brain to help control physical movement and emotional behavior. Increased dopamine triggers neuronal apoptosis. Hyperdopaminergia is a potential mechanism for the inactivation of cell proliferation. Terms to know. AKT is a regulator that promotes cell survival and proliferation and plays a critical role in the apoptosis of neurons in several models of neurodegeneration. Vagotomies is a surgical procedure that removes a portion of the vagus nerve. Haloperidol, antipsychotic medication that works on the brain to treat schizophrenia. It rebalances dopamine to improve thinking, mood, and behavior. Disbidin 1 impacts human cognitive abilities and has been associated with risk for schizophrenia. Reduced disbidin have been reported in the hippocampus prefrontal cortex of schizophrenia patients. Mice that were ventilated show significantly elevated immunoreactivity for cleaved PARP1 in the hippocampal area after 90 minutes compared to non-ventilated mice known as sham. These results indicated that positive pressure ventilation activates apoptotic pathways in the hippocampal neurons. Mice were randomized to high positive pressure alone after pretreatment with haloperidol, an antipsychotic medication, or by bilateral cervical vagotomy, removal of the portion of the vagus nerve. Changes observed after high-pressure ventilation were prevented with haloperidol and vagotomy. No apoptotic neurons were observed. Mice were treated with sham, a type 1 dopamine receptor blocker, and a type 2 dopamine receptor blocker, and then ventilated for 90 minutes. These findings confirm that observed effects are caused by the central release of dopamine acting through type 2 dopamine receptors. This is a representation of the mechanisms of cell apoptosis induced by dopamine. On the left side is the normal conditions of dopamine activating type 1 receptors. On the right side, type 2 dopamine receptors triggers apoptotic pathways that end in apoptosis of the cell. Mice ventilated with either high or low tidal volumes show elevated cleaved PARP1. in the hippocampal area after 90 minutes, compared with non-ventilated mice. AKT GSK3 beta pathway has a huge role in cell survival after cell death. Ventilated mice showed a significant decrease. Increased dopamine levels and activation of dopamine type 2, along with ventilator-induced lung injury, both increased apoptosis and decreased survival pathways causing apoptosis in the hippocampus. Desbiotin levels increased in the hippocampus of mechanically ventilated patients. Afferent signals from the vagus nerve may play a fundamental role in brain injury during mechanical ventilation. Take-home message. Delirium in ICU patients poses serious risks apart from increased mortality, notably brain damage, and what are likely to be related long-term impairments in cognition, mood, and daily living activities. While progress has been made in its treatment, delirium remains common in mechanically ventilated patients. We recently addressed this knowledge gap by studying mechanical ventilation experimentally in mice to determine how it can cause brain damage and thereby the cognitive deficits expressed as delirium. Apoptosis induced by mechanical ventilation is not associated with hypoxia, oxidative stress, or inflammatory responses, but appears to be driven instead by abnormal vagal to dopaminergic neurons that trigger apoptotic signaling via dopamine receptors. Questions after reviewing this article. What does this article say about delirium in critically ill patients? How does this information relate to respiratory therapy in the patients we treat? Is there a possible solution that we as RTs could do to help mitigate hippocampal apoptosis?